Hi everyone, my name is Keyan Peterson. I'm a fourth year medical student at Wake Forest School of Medicine. This is a project I have developed with my mentor, Dr. Stacy Wolf, looking at the contribution of RAS signaling in the development of sporadic cerebral AVMs. This work is supported by the Wake Forest Department of Neurosurgery as well as two medical student fellowships. AVMs are high-flow vascular malformations that are a leading cause of hemorrhage in both pediatric and young adult populations. The vast majority of AVMs, greater than 90%, arise in those with no family history of vascular disease. Recent advances in next-generation sequencing are revealing genetic errors that appear to be present within endothelial cells of sporadic cerebral AVMs. These genetic errors represent a paradigm shift in our understanding of the disease. We have multiple studies that have revealed a high prevalence of mutations in KRAS and the downstream RAS-MAPK signaling axis within endothelial cells collected from human sporadic cerebral AVMs. Of note, these mutations are not found in the paired blood collected from these patients, indicating that these are somatic errors detected only within the lesion itself. The RAS-MAPK signaling pathway underlies cell growth and proliferation throughout our body's cells. It's a highly conserved pathway, and dysregulation of it in other cell types is a known promoter of tumorigenesis. The sporadic mutations that have been identified are in RAS and the key proximal signaling components of the RAS-MAPK signaling axis. They appear to upregulate this pathway and locally promote angiogenesis. The question then becomes, are these mutations incidental or are they causal of AVM formation? This is the key question our research aims to answer. One reason to suspect causality lies in the fact that there are AVM syndromes that occur due to germline mutations in proteins that specifically regulate this same pathway. When these mutations are passed on, these patients develop AVMs ubiquitously throughout their body tissues. For example, in CMAVM syndrome, an inactivating mutation occurs in either RASA1 or Ephrin B4. Whose gene products are negative regulators or basically tumor suppressors of RAS signaling. When these are mutated, RAS signaling is allowed to continue unchecked, much in the way that the gain-of-function mutations in these sporadic AVMs results in upregulated RAS signaling. This shared dysregulation of the RAS-MAPK signaling axis appears to underlie the formation of both sporadic and syndromic AVMs, suggesting that overactivation of this pathway may contribute to the common AVM phenotype. The importance of identifying these mutations in sporadic AVM tissue lies in the fact that this pathway is known to be overactivated in many cancers, including lung, pancreatic, and some melanomas, and it is targetable with well-tolerated chemotherapeutics that may alone or in concert with radiosurgery predispose these lesions to obliteration. This may be especially important for unresectable or residual lesions. The goals of our research are to understand the influence of KRAS and the RAS-MAPK signaling axis on the development of sporadic cerebral AVMs, and two, to create a translational animal model that can be used to test small molecule inhibitors of this pathway with and without radiosurgery. Current animal models are limited in their focus on germline inherited syndromes or by the use of hemodynamic flow models that lack any underlying genetic abnormality. One example of a recent model in rodents came from a group that used HRAS overactivation throughout rat endothelial cells. They were able to produce AVMs in the brain, but this technique was limited because RAS was active throughout the entire body. These rats developed pulmonary and cardiac AVMs, which ended up hemorrhaging and resulting in early death. Further, this model is less translational because the HRAS isotype has not been isolated from human AVMs. We are aiming to create a human representative model in mice in order to have a sustainable translational model for testing of therapeutics and radiosurgery in vivo. So the question becomes, how do we activate KRAS in the cerebral endothelial cells alone without activating it in the rest of the tissues? The answer comes in the form of a newly discovered adeno-associated virus serotype labeled AAV2-BR1, which exhibits tropism or specificity for the microvascular endothelial cells of the brain. So we decided to use this AAV for our experiment. The decision then was whether to attempt an overexpression model by flooding endothelial cells with mutant KRAS, or to create a model by breeding mice that harbor inactive KRAS and utilize Cre-Lox recombination to selectively activate an endogenous mutation. We elected to choose the second model because we felt it more reflective of what we see in human AVM specimens. So our hypothesis for this experiment was, is selective KRAS G12V activation in the cerebral endothelial cells alone sufficient to induce formation of AVMs in the adult mouse brain? There are three main steps to the experiment. First, we obtained lock-stop-lock KRAS G12V mice and built a colony of these mice that harbor an inactive KRAS mutant. Then we injected these mice with the AAV packed with iCre. Again, this AAV only targets endothelial cells of the brain, and the iCre serves to turn on the mutation in wherever it is delivered. So we activated the KRAS mutation only within the endothelial cells of the brain. Finally, we developed a novel method of latex perfusion angiography in order to detect changes in vascular morphology. This is just a brief slide providing an example of how Cre-Lox recombination works. The iCre cleaves the stop site and allows expression of the target gene, in this case, the KRAS G12D oncogene. In our experiment, we had 3 cohorts, 2 experimental in different doses of AAV, and 1 vehicle control. Here's another example of how the experimental methodology using iCre works. In the experimental group, on top, injection of the AAV packed with iCre allows for expression of the KRAS oncogene within endothelial cells, while the control group's expression remains turned off. A brief note on our method for detecting gross changes in vascular patterning. We developed a novel protocol for latex perfusion, which involves traditional injection of liquid latex for casting of the arterial tree. However, for processing, we immerse the brains in scale A2 solution, which allows for rendering the brain's parenchyma transparent and also allows for subsequent immunohistochemical staining. This is a low-cost and effective way to reconstruct the arterial tree ex vivo. So, we allowed for expression of the KRAS oncogene for 2 months in adult mice. Our results demonstrated areas of vascular tangling and altered morphology in 5 out of 12 experimental mice and 0 out of 3 control mice. The arrows point to areas of vascular aberrancy. The final photo is from a control animal. We did not detect any clear AVM-nitis at 2 months. So, what have we learned? Well, our experiment answered one scientific question, that is, endogenous activation of the KRAS mutant alone in adult mice after 2 months does not ubiquitously produce AVMs, but may be associated with arterial tangling or pathologic AV shunting. We need to assess the length of time we allow the mutation to remain active, and we need to also assess the effect of the mutation induction on younger mice. We also developed a novel latex angiography protocol for detecting altered morphology and allowing for subsequent IHC of latex-casted tissue. Going forward, we are currently testing the induction of this mutation in neonatal and juvenile mice from our colony to assess whether timing of mutation onset affects the development of AVMs. We are also looking into using a 2-hit method to assess whether traumatic insult prior to mutation induction is necessary for the development of AVMs. We are using a stereotactic injection to create microvascular insult to the brain parenchyma, followed by a high-dose local injection of the AAV to the frontal lobe to assess for focal AVM formation. This also has the benefit of localizing any lesion to a consistent area for targeted treatments. Finally, we will use this model to test the effect of small molecule inhibitors of the RAS signaling axis with and without radiosurgery to speed obliteration of these lesions. Thank you for your time and for the opportunity to share our work.